Okay, good evening, everyone. So after a long time, we are now going to organize one webinar, and today is the day for it. And it's in silico work. It's an in silico work basically focused on screening of natural alkaloids from Stephania glandulifera Myers, and it's a local plant available in Nepal. And let's explore with its detailed study. And let me share the screen first. Okay. Okay, let's get started. So first of all, let me introduce you to the acetylcholinesterase enzyme. Actually, in this in silico work, we have targeted acetylcholinesterase as a main receptor protein, and actually there are 5 different alkaloids as an inhibitor, or you can say a ligand molecule, to perform molecular docking and further molecular dynamic simulation. And acetylcholinesterase, it is a serine hydrolase enzyme that hydrolyzes the acetylcholine into acetate and choline. Actually, acetylcholinesterase is a kind of carboxylesterase enzyme as well. And let's talk about the acetylcholine as a neurotransmitter. So actually, it's very important in different kind of neurodegenerative diseases. So acetylcholine, it's a neurotransmitter that is found in cholinergic neurons, and it is formed by the combination of choline and acetyl coenzyme A actually, and this is hydrolyzed, or you can say this is mediated through an enzyme called choline acetyltransferase, and later on when acetylcholine is formed, it is further hydrolyzes into choline and acetate by acetylcholinesterase. So in case of neurodegenerative disorders such as Alzheimer's disease, this cholinergic neurons are damaged, and then it leads to the reduced acetylcholine synthesis. And so whenever we are designing a drug against acetylcholinesterase, then these inhibitors block the function of acetylcholinesterase. That means it will increase the level of acetylcholine, and then due to the increase in level of acetylcholine, the sign and symptoms of neurodegenerative disease such as Alzheimer's would be reduced. Okay, in this way the function actually works. And you can see the role of synaptic transmission, as I've already told you about the hydrolysis and all that thing in case of acetylcholinesterase role. And at the right hand corner, you can see the cholinergic hypothesis that was given in 1980s, and it is still relevant for drug designing or drug discovery in neurodegenerative disease. You can see cholinergic hypothesis. It gives the basic idea about the inhibitors that actually target acetylcholinesterase, and it leads to the ACS, that means acetylcholine deficiency, and it leads to the cognitive decline. And there are different drugs that has been approved by FDA, such as donepezil, galantamine, and ribastizine as a reversible inhibitor. Actually, these reversible inhibitors are just a temporary solution. Okay, we don't have any permanent solution, and it's just a hypothesis as well. And although it has been followed for a long time, and it's the only you can say only thing that has been working for designing the drug related with you can see acetylcholinesterase, or you can see this cholinergic neurons. So that is what is going on for Alzheimer's races. And next is, in case of acetylcholinesterase, there is a catalytic triad located in the enzyme, and it consists of 3 different amino acids: serine, histidine, and glutamate. And actually we target these catalytic triads to get our solution. Okay, so that is a basic information about acetylcholinesterase. And let's talk about the natural acetylcholinesterase inhibitors and alkaloids. As I've already mentioned about some of the inhibitors, and apart from donepezil and ribastizine, galantamine is another drug, and galantamine is the only drug that has been approved by FDA, and it's from a natural source, that is a natural alkaloid, and it has been isolated from snowdrop plant, and it is clinically approved. I've already told it. And then it has another role as well. So after after the release of acetylcholine, it can bind onto nicotinic receptor or muscarinic receptor. So it has also a function of binding, or you can say modulating the nicotinic receptor as well, apart from inhibiting acetylcholinesterase. So it has a dual function of being a potent inhibitor of acetylcholinesterase. And apart from that, there are a lot of alkaloid molecules, and alkaloid, as we all know, that it is a nitrogen containing compound, so, or you can say nitrogen rich molecules, biomolecules, and because of that nitrogen containing molecules, it can strongly bind to acetylcholinesterase active site residues. So in that case, alkaloids are very important in neurodegeneration. And next thing is alkaloids do have a broad spectrum property. You can see in the figure as well, there are a lot of bioactivity that has been found to be noted or found to be published in various reputed journals as well. So there are multiple bioactivities, and this because of this bioactivity, they are also considered as a potent inhibitor of particular you can say acetylcholinesterase in this case. And next is at the right hand side, you can see the mechanism of action that I've already discussed, and let's focus on the catalytic triad. So in case of acetylcholinesterase, there are three amino acid residues: serine, histidine, and glutamate. And when we design a drug, and if we get some kind of innovation in this, at least one of them, then it would be easier to inhibit the activity of acetylcholinesterase, and then the cholinergic neuron will get a temporary communication. The temporary communication will be established between the neurons, and then the cognitive symptoms will be decreased. Okay, in this way we can design a drug. And then next is this kind of drug, for example reversible inhibitors. They are also considered as temporary drug, and they can only treat a mild to moderate kind of Alzheimer's disease. Okay, so it's not a complete solution actually, but it has been used to against some of the common symptoms and signs and symptoms shown by patients of Alzheimer's disease. Okay, and next is, so actually in this research we have used Stephania. Stephania specifically, there are different species of Stephania, and Stephania glandulifera was particularly used. Later on I will let you see the plant body as well. The flower is presented over here, and it is very important to taxonomically identify this species as well. And this Stephania glandulifera, it belongs to the Menisperci family of Angiosperm, and it is one of the largest family of Menisperci. It is one of the largest genera of Menisperci actually, and it is a rich source of alkaloid. Various groups of alkaloids are present in this particular species or particular plant. And in case of let's talk about let's talk about the species of Stephania. So there are different species. Among them, in our study, we use Stephania glandulifera. Apart from Stephania glandulifera, apart from glandulifera, so there are other species as well that are prevalent in this region. Stephania glandulifera, Stephania glabra, and gracilenta. And the main distinguishing point of this species is the flower. Okay, flower itself, the inflorescence and all that thing get them distinguished into different you can say species. So that is how we taxonomically distinguish Stephania glandulifera, and it was extracted, or you can say it was found in the altitude of 1500 meter in the eastern part of Nepal, in the Ilam district of Sri Antu. And the major compound that we successfully isolated and elucidated were Stephania, stephalidin, stepharanin, tetrahydropalmatin, and palmatin. They were isolated from this particular species, and the plant body used was caudex. Actually, caudex is a modified stem present just above the swell of the plant habitat, and performing different methods, for example column chromatography, high performance liquid chromatography, and you can see fractionation as well, and then the cold percolation method was used to obtain the alcohol extract, and then the alcohol extract was fractionated in different solvent extract, and then with each of the solvent extract column chromatography, repeated column chromatography were performed, and then finally taking those fractions, we performed preparative HPLC as well, and then using that preparative HPLC we isolated these compounds, and then further 2D NMR and then the mass spectrometry was performed to elucidate those structures. And thanks to different personnel involved in this research, I will acknowledge them at the end of this presentation. Okay, so and it has been already published as well. You can see the citation over there in 2023. So let's move ahead. So there are actually 5 different alkaloids isolated from this species, and these alkaloids falls under different you can say group of alkaloids. For example, in case of stepharin, it has a different structure other than those 4 molecules. This stepharin is a class of proaporphin alkaloid, and then tetrahydropalmitin, okay, THP, tetrahydropalmitin, and stepholidine. So and these falls under tetrahydroprotoborburin. Okay, tetrahydroprotoborburin. 4 hydroxy group are present over there, and the next is palmitin and stepharin. Okay, so palmitin and stepharin falls under protoborburin class of alkaloids. So we have isolated 3 different classes of alkaloid from a single plant, and this work of isolating these 5 alkaloids from Stephania glandulifera has been done for the first time around the world. Actually, it's not a novel compound, but we have isolated these compounds from Stephania glandulifera for the first time. Okay, and in this in silico work, the main objective of this work is to evaluate these 5 alkaloids. As an inhibitor of acetylcholinesterase to be taken into further drug designing, or you can say taken into further clinical trials as well as in vivo work in respect to Alzheimer's or any other neurodegenerative disease, particularly Alzheimer's. Okay, so these are the basic research objective of our work. So and let's talk about the methodology now. So we have performed molecular docking, molecular dynamic simulation, and in silico pharmacokinetics to screen these compound molecules, and then we got the conclusion, and then we can also do further works. We can share that later on. Okay, now let's talk about molecular docking. So in case of molecular docking, as I've already told you, we selected the acetylcholinesterase receptor, and then particularly we selected acetylcholinesterase with a PDB ID of 1 QTI, and it was extracted from an electric ray that is Torpedo californica, and then because of the homology similarity, or you can say because of the similarity of this sequences with the human, we selected the Torpedo sequences over there, and then we perform molecular docking, and then molecular docking was performed through Autodoc Bina, and then we use Autodoc tools as well for the preparation of molecules, and then we use Discover Studio Visualizer as well as Chimera for visualizing and energy minimization, all that thing. And then you can see the grid box dimension and size as well. And then later we selected the best conformation, and taken into that conformation we perform molecular dynamic simulation using Mastio, a version of Schrodinger suite, and then we perform MD simulation at 200 nanosecond. Actually, we performed MD simulation in 100 as well as 50 nanosecond as well, and we evaluated everything, and then at 200 nanosecond it was up to 200 nanosecond it was quite stable. Okay, and then we use Olive's 2005 force field, and then T3 solvent molecules, solvent system, you can say a water solvent system over there, and then there's these force field were used to calculate the forces and energy to stabilize that molecule, or you can say optimize the molecule, and solvent model, as we all know, we simulate the similar system of human body into the in silico work. So we just created a solvent box, that is a water box, and then added you can say ion molecules, 0.15 molar of sodium chloride, as well as as well as the pressure was maintained to 2 ps, as well as the temperature was maintained to 300 Kelvin, and 1 bar pressure was also used. And then the RAS per integer was also used to calculate the bone force, and then the time step in the simulation was used for 2 femtosecond. And then we analyze those we obtain those trajectory, and analyze those, or you can say um, analyze those trajectory to obtain the different graphs and data that we call it post simulation. And then based on the analysis of RMST, RMSF, and all that thing, we'll later talk about it. um, Based on all of the things, we concluded the result. And later we also performed in silico pharmacokinetics. Actually, in silico pharmacokinetics was performed before molecular docking. Actually, in silico pharmacokinetics, we perform through Swiss ADME as well as PKCSM. Several other software were also used, but these are the two best software tools for in silico pharmacokinetics. Actually, Swiss ADME was used to analyze the drug likeness of this alkaloid molecule, whereas PKCSM was used to analyze the broad spectrum, or you can say broad spectra of ADMET parameters. There are a lot of other parameters involved here. We'll discuss about it later. Okay, now the workflow of drug design for this particular, you can say, drug screening for this particular research work. So first of all, we primarily screen the compounds. For that, we isolate the compound, and then later we elucidate those alkaloids, the structure of those alkaloids, and then characterize the compounds. And then further we prepare those alkaloids for molecular docking. Here there are a lot of steps involved in preparation of ligand molecule, actually energy minimization, and then adding a force field, and then simple tasks that you can say charge addition, nitrogen addition, and all that thing was done. And then later on we perform molecular docking. For molecular docking, we also selected the appropriate receptor for this alkaloids, and it was acetylcholinesterase. I have already told you 1 QTI is a PDB ID of that receptor. And then we also predicted the binding site using PDB-SOM, CASP, and a lot of things, a lot of online tools. And then we perform molecular docking with Autodoc Vina, and then we selected the best conformation out of 9 different conformation. And then based on the best conformation, we perform for the molecular dynamic simulation. And in within that process, we also validated our docking results. Later on we will talk about the validation as well. And the next is we perform molecular dynamic simulation, taking the best conformation, or you can say best complex molecule, actually the conformation along with the protein. And then we set up different parameters for molecular dynamic simulation, and then we run that simulation, and then we obtain different trajectory, and then based on that trajectory we analyze them, and then check the stability of the ligand molecule into the protein with that basic conformation. And then we also calculate the binding energy, minimum binding energy, through MMGBSA. And then we also calculated the flexibility, or you can say conformation of the protein molecules through PCA. Principal component analysis was done. And then later we also perform in silico pharmacokinetics, the drug likeness, as well as ADMET parameters. Various other ADMET parameters were analyzed. So it is the workflow of our research work. And then let's move on to the result section now. So molecular docking results. So in molecular docking, we obtain different conformation, and the best conformation has been presented over here. In case of stefarin, the best conformation, so the binding affinity of minus 10.3 kilocalorie per mole is a very best binding energy. And then there were 2 hydrogen bond hydrogen bond residues, and then the length of hydrogen bond, you can see it's very low. You can see the guideline value is 3 angstrom. Above 3 angstrom we can consider the hydrogen bond as a conventional hydrogen bond. So both the residues has hydrogen bond length of less than 3 angstrom, so they are good. These are good results for stefarin. And then the hydrogen bonded residues are were tyrosine 121 and histidine 440. Actually, histidine 440 is present in the catalytic triad among the serine and glutamate, and it is the best result out of these 5 alkaloids. And you can see the binding affinity of other alkaloids as well. Although they have a good binding energy, because of lack of interactions, we didn't select other 4 compounds as a probable molecule to screen for them. So these are the results for molecular docking, and you can see the best conformation and the interaction as well. Here you can see at the left hand side staphyrin interacting with the key residues histidine 440 in the catalytic triad, and apart from that tyrosine 121 is also another residue that has a hydrogen bond over here. And then you can see other interaction as well, other hydrophobic interaction, tryptophan 84 and several vanillus interaction. And then these are the basic. Okay, and next is tyrosine 121. Although it is not in catalytic triad, it is present in the active site, and it also provides extra stability for the molecule. And it is a docking validation, and docking validation was done in 3 different steps. Actually, I have only presented the first one, and a very vital validation result as well. So in case of docking validation, we actually redock the parent ligand. The parent ligand is galanthamine, so we dock the galanthamine molecule, redock the galanthamine molecule into the active site, and then select the best binding best conformation. Actually, they selected the best binding conformation, or you can say the best conformation in the binding site, and then we superimpose the crystallized one. That means the original structure and then the best poses, and then we calculate the RMST. So actually, after performing this superimposition, we got RMST value of 0.9 angstrom. So that is that is actually a good RMST value, and it shows the accuracy of docking as well. You can see in the figure as well how it has been perfectly aligned with each and every residues, although there is some difference at the end, or you can say at the back end of this molecule, but it is acceptable. It is acceptable in case of molecular docking. And let's talk about the Lipinski rule of 5 or drug likeness of this different ligands. So there Lipinski rule of 5 actually is widely studied or widely validated some kind of drug likeness model present over there. Then there are 5 different parameters that is given over, and then any kind of molecules, any kind of compound molecules or ligand molecules, if they are considered to be a drug like nature, then they need to fulfill those parameters. For example, molecular weight that needs to be less than 500 Dalton, and then hydrogen bond donor, it needs to be less than 5, hydrogen bond acceptor, it needs to be less than 10, log p value less than 5, and molar refractivity, it needs to be it needs to lie from 30 to 140, you can say. So actually, when we analyze the result of these 5 different compounds, you can see all of them have fulfilled the parameters. However, staphyrin has been the best after analyzing the broad spectrum of ADMET parameters. Okay, and these log p value and molar refractivity, they are very important in solubility of drug as well. And let's talk about the broad spectrum of ADMET parameters. So in case of ADMET, the broad you can say the quantitative analysis of ADMET parameters, we use PKCSM, and then it calculates different parameters in ADMET. For example, in case of absorption, there are different parameters: water solubility, cathode permeability, intestinal absorption, a lot of things are there. And in case of distribution, volume of distribution, fraction unbound, blood-brain barrier, CNS, and all that thing are present. And in case of metabolism, cytochrome is very important player in this metabolism, cytochrome 440 actually, and there are different isoform in cytochrome 440 and 450 actually, and we have analyzed this PKCSM usually analyze these isoforms as well. And after that, in case of excretion, that is total clearance, and renal OC2 substrate are there, and then toxicity for toxicity AMS, and a lot of other things are involved there. And now you can see I haven't mentioned the guideline value over there, but all of the you can say the values are falls under the guideline value. And in case of let me explain some of the vital parameters. For example, in case of water solubility, it is water soluble, that directly links with the solubility of the rock. And then you can see in case of glycoprotein, actually glycoprotein, you can see glycoprotein means it's a carbohydrate containing protein molecule, and then it is very important for drug efflux pump or drug efflux mechanism as well. And if a drug molecule is considered to be a substrate of glycoprotein, then it directly impacts the oral bioavailability. It decreases the oral bioactivity. So if you see in case of stepharin, the substrate nature is not present, or you can say stepharin is not a substrate of glycoprotein. Okay, so that means it has a good bioavailability, or you can say solubility. And being in case of inhibitor as well, if it is an inhibitor, then you can say it can lead up to toxicity. For example, drug-drug interaction or unspecific binding can occur in this case. So these also looks fine as well. And let's talk about the metabolism now, where cytochrome is present over there. As I've already told you, cytochrome is a very important player, or you can say a central player in drug metabolism particularly. And there are different isoforms of cytochrome 440, and they have the significant role, different role in drug metabolism, particularly cytochrome A4. Okay, where is A4? Okay, cytochrome 3A4. It is very important isoform that in this case the database has analyzed that for 50% drug molecules, they gets metabolized through their cytochrome 3A4. So it is very important as well. So if a molecule is a, or you can say if it is a substrate, then a drug is easily metabolized by the enzyme. So you can say cytochrome A3, it is a substrate. Okay, so that means it is easily metabolized in the liver as well. And if it is a inhibitor, then you can say inhibitor easily blocks it. Okay, so there is no different there. There is one inhibitor case, cytochrome 2D6. So cytochrome 2D6, it has a polymorphic property, so we need to broadly analyze in vivo as well later on, so that we can actually say that if it actually inhibits or not. Okay, and then it also affects the drug response. So it doesn't have a particular role in, you can say, a basic role in drug metabolism in this case. Okay, in this isoform. So and later on you can see in case of hepatotoxicity, it is said that stepharin has a hepatotoxicity nature. Okay, actually in case of alkaloid, they have a trace amount of hepatotoxic property that you can say it can hamper the liver as well. But we need to broadly analyze what is the quantitative measurement of being a toxic nature. Okay, so if it is below the guideline value, okay, we can just analyze that through in vivo. After performing in silico, we can go to in vivo, and then later on we can go to trial. So before going into trial, you need to go through in vivo models as well, mouse model or whatever model. So in that condition, we can easily study the hepatotoxicity nature, what is the particular value that is to be considered as a hepatotoxic nature. So that is only the thing that we need to do for admit parameters analysis of this particular drug. Apart from that, I haven't explained in detail about palmitin, palmitin, stepharin. You can just check that figures. And now let's talk on MD simulation. So in case of MD simulation, as I've already told you, we have performed MD simulation through Maestro, a Schrödinger suite platform, and then we did molecular dynamic simulation for the best conformation only. Okay, the best conformation of stepharin, tetrahydropalmitin, or you can say palmitin, and other drug, or the five other drug, those who were structurally elucidated. So let's talk about the first figure. So it is an interaction map, or you can say interaction of staphyrin in the ACHA receptor protein, and the A implies before simulation, and B implies for the after simulation confirmation. Okay, so you can see there is no such deviation or no such conformational change in the ligand molecule or staphyrin present in the active site of ACHE. And then we calculated the RMST as well. The RMST root mean square deviation was just 0.9. That means it has not been you can say changed over simulation 200 nanosecond simulation time period. And in the middle of this slide you can see the RMST plot, and in this RMST plot the red you can say the red figure, it illustrates the RMST of ligand, and the blue one, it signifies the protein RMST throughout the simulation up to 200 nanosecond. Actually there has not been too much fluctuation, but you can see a slight deviation up to 25 angstrom. Okay, and in case of molecular dynamic simulation you can expect this result. Okay, in the starting of simulation drug needs to get into that particular rhythm, so it needs some time to be particularly active over there. So it's a kind of you can say a basic preparation thing up to 25 nanosecond, and after 25 nanosecond it forms a stable curve, and it in this figure as well you can see there has not been too much deviation. Okay, and then later on after 175 you can see in case of ligand RMST there are some fluctuation, and after noticing these things we can just compare the residue index and all that thing through the results obtained from RMST. And after seeing that we found out that the fluctuations were away from the active site residues. Okay, or you can say the fluctuation were shown by the residues that were away from the active site. That means it has not changed over the time. Okay, that means the figure is quite stable. And at the corner right hand side you can see the RMSF plot, root mean square fluctuation of 1 QDI with the staphyrin, and it also shows the stability of protein molecule, you can say stability of complex molecule, where protein and the particular residues particular you can say ligand molecule has been interacted over there, and you can see some great spike present over there in this particular region. There are 3 different spikes present over there. However, these spikes okay, let me tell you a idea of this. Okay, the residue index, that implies the residue count of amino acid. Okay, actually it is not necessary that, for example, in case of 110 or something like that, there is one dark green color. Okay, green color pictorial representation over there. That doesn't mean it is the amino acid with a number of 110 or something like that. Okay, it is quite closer to it, but we need to analyze the figure over there. Okay, trajectory analysis needs to be done to analyze actually what kind of residue is present over there, or you can say you can just compare the residue index with your residue number over there. Okay, amino acid number. So in case of this figure, the dark green coloration that denotes the key residues that has been involved into the you can say interaction with the ligand molecule. Okay, so particularly we have histidine 440. So it has a fluctuation RMSF value lesser than you can say 0.5. so it is quite stable. And what is that? Next is 84. Okay, 84, that means this one is for tryptophan, although it is not clearly mentioned over there. When we analyze the data, it didn't show the RMST of 1 angstrom. So the average RMSF of different residues present over the time frame were just 0.5 to 1 angstrom. That means it is quite stable as well. And looking at the spike, these spikes occur due to some kind of conformation, you can say the protein flexibility, and it was not even the key residues involved, so it doesn't hamper the result. And next is SSE plot. That means it is a protein structure. You can say the secondary structure elements has been analyzed over there. It particularly analyzes the protein structure integrity. You can say the stability of protein. And then in the top hand side, you can say percentage SSE, and then you can see a graph, stable graph present over there. That means the residues are quite stable over the time of 200 nanosecond. And then on the later on the later graph, or in on the downside, you can see two different you can say the color notation. The red color, it signifies is the alpha helix of the protein, and then the blue indicates the beta sheet. Okay, and then you can see the dark line, it indicates the greatest stability of the residue molecule. You can see most of them are stable. And then we just need to see the key residues. Okay, the key amino acid residues that have been interacted with the ligand molecule. So all of them are quite stable over here. And then next, in the middle, you can see a contact map of different amino acid residues with the ligand molecule. You can see the basic, you can see the vital amino acid. For example, histidine 440, that is a hydrogen bond. The green coloration denotes the hydrogen bond, and it has not been deformed from the molecular docking to assimilation, as I've already shared the interaction pattern in molecular docking, where histidine 440 was interacting with the ligand molecule, forming one hydrogen bond, so that means it is not deformed, it is stable throughout the simulation. And then next is tyrosine 121. Although it has a the level of hydrogen bond is quite low, but still it has been preserved. And next is tryptophan 84. You can see a good interaction contact map, or you can say a good contact map of the interaction of tryptophan 84 with the amino acid. So it just signifies how much residues has been deformed or how much has been conserved. Okay, that means not only most, all of them are conserved in molecular dynamic simulation. That means the molecular docking was performed quite you can say effectively, or you can say the results were quite good. And then next is, you can say there are various plots given over there. RMST, we have already discussed about the RMST. Next is the radius of gyration, and this radius of gyration, it measures the compactness, or you can say the size of molecule in the simulation, and it also gives the average distance of all the atoms from the molecule from the center of mass. Okay, or you can say center of molecule. And the low radius of gyration, it directly signifies the molecule is more compact. And then the higher value, or you can say the more the non-stable curve, it gives the molecule has been more folded. Okay, more expanded as well. In this case, you can see the stable curve has been obtained over there. Although there is some minor fluctuation, that is not the point of concern. Okay, and then next is the SASA plot. Okay, SASA plot, that means solvent accessible surface area. It indicates or you can say it measures the surface area of molecule that has been exposed to the solvent. In this case, we have used water molecules, or we always use water molecules. And then the results, you can see it is quite unstable up to 125 nanosecond, and later it gains the stability up to 200 nanoseconds. So that means the protein has some kind of you can say confirmatory change to 125 nanosecond. So apart from that, there is another parameter that is called mole assay, that is molecule surface area. You can see a plot, it is quite stable. Actually SASA and mole assay are quite similar, but in case of mole assay, it measures the interaction of molecule beside the solvent. Okay, beside the solvent there are other different residues as well, so it measures the interaction of other residues. In case of SASA, it is only concerned with the water molecule, or you can say solvent molecule, but in case of mole assay, it is concerned with overall different other residues as well. So it is quite stable. So overall, if we analyze these overall parameters, then it is quite stable in case of stepharin. So I have only presented the result of stepharin. Okay, stepharin with 1 kDa. And next is intermolecular hydrogen bonding. So we didn't detect any kind of intermolecular hydrogen bonding, or you can say intermolecular hydrogen bond. In this case, if we see any kind of intermolecular hydrogen bond, then that can make the molecule unstable, or the unspecific binding can also occur. So in this case, the result is quite you can say satisfying as well. So now let's talk about mmgbsa binding energy. So mmgbsa binding energy, so we calculate the minimum binding energy after the simulation as well. Okay, so before the simulation we do it through molecular docking, we calculate the binding energy, or you can say binding score, and after molecular dynamic simulation we just need to calculate the minimum binding as well, binding energy as well, to see whether it has been bounded with the same kind of efficiency or not. Okay, so in case of stefarin, although the binding energy looks different, but if you talk about the overall binding energy, it is quite low. Okay, but if you compare with rest of the four, it is quite larger as well. Okay, but if you compare with overall the guideline, or not guideline actually, if you compare the overall structure, then it is quite low. So it has a good binding affinity. And now let's talk about the PCA plot. Okay, PCA analysis, principal component analysis. It is analysis that particularly does the work for say alpha atom of protein. Okay, and then you can say different coloration denoted for different complexes. For example, blue color, it denotes for palmatine complex, and then orange color, it denotes for stefarin. You can say gray color is for stefolidine, SPO, and then stefarin for orange, yellow, and then THP, tetrahydropalmatine, for green coloration. And it gives the dynamic behavior of molecule actually. So if you see the green coloration, it is present in the central region, and it is more conjugated in that region. So it looks like it is in this case tetrahydropalmatine complex has not have that kind of flexibility. Okay, but in case of a stefarin, you can see it has been scattered through different quadrants. Okay, it has been scattered into 3 different quadrants, not only 3, 4 as well. So it means the protein has a great conformation flexibility, and then it doesn't mean that it is unstable. Okay, because we have already proved from RMSF as well as RMST and different other plots, so we have already concluded that it has a quite good stability. Okay, so PCA just look about the flexibility of molecule. Okay, the flexibility can occur not only through the active site but away from the active site as well. So it calculates overall structure. So because of those spikes, maybe because of those spikes, in case of PCA, it has shown a great movement. Okay, so this is all the result of PCA. And lastly, after analyzing all the results from molecular dynamic simulation, molecular docking, molecular dynamic simulation, and ADMET, we came to the conclusion that this stefarin molecule has a great potential to be a good inhibitor. Okay, you can say potential inhibitor for acetylcholinesterase, to find a new molecule, to find a natural molecule as well, natural alkaloid. After galantamine, we haven't seen any kind of drug molecule that has been approved by FDA, and this molecule can provide that alternative as well, provided we need to do this work in the advanced level as well. For example, in vivo as well as in vitro work needs to be performed first, and then in vivo work needs to be performed, and then we can go through if everything goes fine, then we can go through clinical trial as well. Okay, so based on this research, we have concluded that stefarin has a great potential to be a Alzheimer's drug as well. And okay, we have not just look for the docking, we not just look for the simulation and ADMET as well. We combine each and every results of 5 different alkaloids and then compare them, and then yielded this conclusion. Okay, and then in case of admit as well, although there was one slight changes, you can say the hepatotoxicity nature was there, although in case of galantamine and donepezil as well, we found that they have some hepatotoxic property. So maybe because of alkaloid nature, it has some kind of there's a negative response in the admit, but we cannot conclude in that way. So in further research we can easily identify what is the you can say what is the value that is giving it to be as a, you can say, better toxic nature. So we need to study in the deeper level. And this work, this whatever I have presented over here, this is based on the two major research work in Stephania glandulifera. You can see a plant over there. It is a vine, and it consists of tendrils, and I have only shown the odd-shaped leaf of Stephania over here, but actually we didn't use the leaf. We use Caudex modified steam pat, and it varies in size. We can get over 20 or 30 keys as well in a single cortex. So we have used cortex for screening. Okay, and um, the previous research was focused on compound isolation as well as structural elucidation of those compounds, and then performing the cytotoxic nature. Actually, this alkali molecules was also found to be active in adenocarcinoma as well, glioblastoma cells, and it was already published in Journal of Biologically Active Product from Nature, a Taylor and Francis journal. And then later on, we performed in silico work, and it has already been published in Journal of Biomolecular Structure and Dynamics in recent March 2025. So this is all about the research work. So I'm here, the presenter. So if you have any queries, or if you have any kind of you can say collaborative proposals or whatever, you can contact me. We can work hand in hand. And next, let's talk about our work as well. In Nepomics Biotech, we primarily focus on workshop and webinars. We have been organized, actually. We have been organizing different virtual workshop on CAD, and especially focused on CAD, but we also organize other programs. We have already initiated Python programming language basis to advance as well. And then we also conduct different other academic work. For example, we have also we have done one workshop based on article writing, research writing. And then apart from this kind of academic works, we also focus on research and development of natural product. We particularly use essential oil for this. We have used essential oil in the past to form the different personal care product. There are 4 different personal care product that has been already formulated and commercially available on marketplace. And there is a contact detail of Nepomics Biotech. If you need any kind of help, or if you want to collaborate with us, then this is it. So thank you for all the attentions, and from coming here for this webinar, and we look forward for any such kind of collaborations, and for the works on plant-based product, or you can say any kind of academic stuffs. Okay, this is it. So if you have any queries, you can arise here. Okay, then I don't think there are any questions. So let's end this webinar. We will meet on another program. Soon we are organizing one workshop that will be a combo workshop of Python programming language, basic Python programming, along with the CAD, a different sort of CAD programs. You can see basic molecular docking, and then uh, homology modeling, basic bioinformatics, all that thing. So we will see, we will organize that program soon as well. So thank you all for the for coming over here, and then